Hi class, my name is Sarah Wilkins. This is the Advanced Group Concepts Presentation 7. Lovastatin, which is also known as Mevacor, is a drug made by Merck Pharmaceuticals and that is used to lower cholesterol. For this presentation today, we're going to kind of research the drug lovastatin a little bit, kind of dig into that a little more. And we're also going to walk through cholesterol biosynthesis. And we'll also talk about just how lovastatin is going to inhibit cholesterol. That is one of the properties. And if lovastatin goes through any structural changes before it becomes active, we will also discuss this during the presentation. And we're going to use a couple of images, kind of look around at that and get an idea. All right. First, the first step here, something that we really want to look at that plays a big role in this that we need to be aware of. This is going to be fatty acid synthesis, and that's the picture that's here on this slide. It shows the steps. You can see the liver and the adipose tissue. We can see the acetyl-CoA, we can see the melanin-CoA, the carbon dioxide, the acetyl-CoA, and then the carboxybiotin, sorry, hard time saying that, and the biotinate. So essentially, what is fatty acid synthesis? Fatty acid synthesis is the creation of fatty acids from the acetyl-CoA and the NADPH. And this creation is it's going to be done through the action of enzymes that are called fatty acid synthesis. So those are kind of the parts that are giving the action to this process that we're looking at. And this process does take place within the cytoplasm of the wall, which we already know because we've learned that. And most of that acetyl-CoA, which is converted into fatty acids, is derived from carbohydrates via the glycolytic pathway. So some of this probably sounds familiar. We have went over a lot of it. I just wanted to walk you through that a little bit. So to talk about lovastatin a little bit, lovastatin is structurally similar to HMG, which HMG is a substituent of the endogenous substrate of the HMG-CoA reductase. So we have to keep that in mind. Lovastatin is a prodrug that is activated in vivo via hydrolysis of the ring, the lactone ring, from the B-hydroxy acid. So, like I said, that lovastatin is a prodrug, and it's an inactive lactone in its native form. The gamma-lactone closed ring, which is how it's administered, is hydrolyzed in vivo in the open ring form, which we talked about. So the hydrolyzed lactone ring is going to mimic the tetrahedral intermediate produced by the reductase, and this is going to allow that agent to bind to that HMG-CoA reductase, and that's going to be done with 20,000 times greater affinity than its natural substrate. The bicyclic portion of the lovastatin is going to bind to the coenzyme, which is a portion of that active site. All right, and here we're going to look at that HMG structure, and I want to look at this HMG structure because of the fact that it's so similar to the lovastatin, right? So the HMG-CoA reductase, that's going to be the rate controlling enzyme of the mevalonate pathway, which we already know plays a role. And we're responsible for cholesterol and other isoprenoid biosynthesis. So HMGR is going to be transmembrane protein. So remember, transmembrane protein, and it has 8 domains. That's going to be anchored in the membrane of the endoplasmic reticulum. So if we look there, we can see how those structures are very similar.